Today we'll be speaking about genetic engineering and how it can lead to the technological singularity. Developments in genetic engineering have this potential due to their ability to create augmented superhumans with enhanced health, intelligence, and other physical traits. However, genetic engineering technologies like CRISPR need to become more accurate and less unpredictable before this can happen. CRISPR is a genetic system that was originally found in bacteria. Today it is used in genetic engineering as a tool to modify specific genes. CRISPR produces an enzyme, Cas9, which binds to a specific DNA sequence. After binding to it, the CRISPR-Cas9 system can modify, cut, or activate the genetic sequence. More specifically, CRISPR works by searching for repeated genome sequences, which it is able to easily identify and latch onto. By looking for a repetition instead of checking each sequence, CRISPR is able to make changes in a genetic sequence in weeks, whereas alternative methods might take months. Furthermore, through the use of Cas9, CRISPR is able to target more than one gene at once. One successful application of genetic engineering involves giving an animal the traits of a different species. Researchers at Craig Biocraft Laboratories were able to splice spider silk proteins into the silkworm genome. The resulting silkworms are able to spin spider silk, a normally difficult to obtain material due to the difficulty of raising spiders in captivity. Spider silk is valued for its strength and elasticity compared to contemporary fabrics, and is widely regarded as one of the toughest fibers on Earth. The existence of these modified silkworms could allow companies to widely use a much stronger thread in many products, such as parachute cords, high-performance sportswear, and suitwear thread, which is what is used for stitches after surgery. Of course, scientists desire to extend the application of genetic engineering to creatures more complex than spiders and worms. Researchers experimented on fertilized rat eggs and discovered that a single injection of CRISPR was able to modify 1 to 4 distinct genes in them. These animal tests are important by setting a precedent for best practices and more experiments.
Using a virus as a vector, Dr. Ophir Shalom and his team inserted the protein into a collection of embryonic liver cells, hoping to delete a DNA sequence responsible for manufacturing a certain protein. After 11 days, they discovered that the targeted protein had been muted in 93% of the cells, as shown on the left. The right side shows an alternate method which clearly was not as effective. After their success with the liver cells, the team used a gecko library. The library contains information on 18,080 genes, allowing the CRISPR-Cas9 system to easily scan human cells for predefined sequences. This makes it simpler to knock out or remove a gene sequence from the DNA. Now, using this gecko library, Dr. Shalom and his team attempted to knock out a DNA sequence in melanoma cells that caused resistance to PLX, a protein that can be used to treat melanoma. By removing the sequence causing resistance, genetic engineering would make melanoma more easily treatable. After 14 days of PLX treatment after the gecko library was used, Shalom found that the melanoma cells were failing to make essential proteins. Unfortunately, not all experiments have been so successful. Genetic researcher Zhang Huang and his team looked to use CRISPR to knock out genes that caused a deadly blood disease. They inserted CRISPR-Cas9 into 86 embryos that could not have led to live birth. However, of those 86 embryos, only 71 survived after 48 hours. At this point, the team tested 54 embryos and found only 28 were repaired by genetic engineering. Furthermore, a high proportion of the embryos showed signs of mutations that were not intended by the experiment. The possibility of these few mutations being harmful to the embryo is concerning enough, but Huang only experimented on part of the human genome. If they had looked to fix the mutations causing the blood disease throughout the whole genome, there likely would have been even more errors. And outside of the failures of this experiment, there are other issues with CRISPR. For example, there's the potential for extraneous DNA to pollute several strands of DNA after CRISPR cuts them, but before they can fully reconnect. When this happens, it can create biologically non-physical strands of DNA that do not function properly, which can have a disastrous consequence for the host. Another issue with CRISPR technology is known as the Energizer Bunny problem. CRISPR requires a delivery system to reach the target cells that need to be modified. The method that scientists use is to put CRISPR into viruses, and then have these viruses deliver CRISPR to the cells. However, the viruses might continue to generate CRISPR for 10 to 20 years, which could lead to unpredictable genetic mutations.
Scientific research is often very expensive, and when a wealthy sponsor is backing a research team, they want to show the companies supporting them that their dollars are not going to waste. In that case, scientists often attempt to portray their data in a more positive manner than what one would normally expect. This can lead to improperly designed experiments and skewed results in the scientific community. At its current state, CRISPR is too unpredictable to use reliably on human embryos. Huang suggests the possibility of first trying it to decrease random mutations in animal models. Then, hopefully, once the technology has been perfected there, it will translate over to embryos. This will allow scientists to ethically conduct experiments to perfect the technology before it can be used practically on humans. The genetic engineering technology that already exists could be applied to humans in the future. Examples of what might happen to humans who are genetically altered are prevalent in science fiction. For example, the Maximum Ride series follows the lives of genetically modified children who were enhanced with bird genes as embryos, which gives them special abilities such as flight and stronger bodies, and turns them into a variety of post-human. One popular science fiction example of a genetically engineered human is Spider-Man. Since genetically modified silkworms that can spin spiderwebs already exist, it is quite possible that a Spider-Man-esque transhuman could be created in the future. However, future applications of genetic engineering could have a profound effect on humanity even without merging aspects of different species. Just modifying the human genome with other human genes could result in the creation of a superhuman. Currently, genetic engineering research is primarily concerned with curing genetic diseases. However, once CRISPR technology is advanced and reliable enough, people might be able to choose what traits they want for their children, both physically and mentally. Genetic engineering also has the potential to be applied in a militaristic fashion as well. In a science fiction franchise known as Prototype, the main character genetically modifies a virus to weaponize it, and once he himself becomes infected by the virus, he transforms into a living weapon. With the ability to shapeshift his limbs into deadly objects as well as a potent regenerative ability, he becomes the epitome of a biological human weapon. If CRISPR ever becomes refined to the point where the previously mentioned scenarios are no longer relegated to the realms of science fiction, then it will be able to bring about a technological singularity via a genetically modified post-human race. A reality with living angels, weaponized humans, and perfect beings could completely revolutionize the human way of life. But until CRISPR's many issues are solved, this future will only remain a dream.